So the next type of media is blood agar, a very commonly used type of agar in hospitals. This is pretty much going to be the first type of agar that every type of or that every culture is going to be started with in the lab. Pretty much everything grows on blood agar. This is nutrient agar plus. This is nutrient agar for fastidious organisms. I mentioned Streptococcus doesn't really like to grow on nutrient agar. It will, it just grows really slow. This is the type of agar that Streptococcus loves to grow on. Okay, so this agar, blood agar, is essentially nutrient agar with 5% sheep's blood added to it. Sterile sheep's blood from a sheep. Sometimes goats are used. They use sheep because of the proteins that are found on the red blood cells. That's the easiest for bacteria to break down for some reason. Other species have different forms of proteins, which bacteria have a harder time breaking down. Okay, so blood agar. It is nutrient agar, which is that color right there, plus blood, giving it the red color. And that'll come back to play here in just a second. So this is a non-selective media. Pretty much everything will grow on this agar that touches it. If it's gram-positive, if it's a rod, if it's a coccus, if it's gram-negative, it will not matter. Pretty much everything will grow on here. This is a type of nutrient agar plus blood, and the blood gives it gives the fastidious organisms what they need to grow as well, which is why this is used for strep. It's a fastidious bacteria. It is not selective. Pretty much everything will grow on this. But it is, however, differential. What does grow on it, if you know what you're looking at and you've done your diagnostics correctly, you will be able to tell apart what you're looking at. Now, this particular agar is designed mostly for Streptococcus species. You can use it for other different types of bacteria, but what happens at this point is you should have done a gram stain in the lab. You looked at it underneath your microscope and you went, oh, this is a gram-positive coccus. You put it on a mannitol salt agar plate at the exact same time as you do a blood agar plate. The mannitol salt agar plate, Streptococcus will not grow on mannitol salt agar plate, but it will grow on your blood agar plate. And that knowledge tells you that it is not a gram-positive coccus, Staphylococcus, since it didn't grow on here, but it did grow on here. It was a gram-positive coccus, which means it's probably some sort of strep. And this is how you tell apart the different species of strep, the differential aspect. So just because it grows doesn't mean it's a strep, but if you've done your job correctly, you will probably only have streps left. And this is where the differential aspect comes into play. Now, the differential media, the differential agent in blood agar is the blood itself, red blood cells, and that's the differential agent in this. There is no selective agent. Pretty much everything can grow, but it is differential. The differential agent in this case being red blood cells. So what happens is you take a sample from your patient and you put it on this blood agar plate. You put it in the incubator. You come back and you look and see what the bacteria did to the red blood cells themselves. Now, this is what a blood agar plate looks like out of the package or when you make it yourself. It's nice and blood red. There's nothing growing on it. You can see that it is nice and solid red. Now, you put your streak on here. You come back and look at it, and after it's been in the incubator for 18 hours, and you see a color change. What you're looking at is what is going to occur to the red blood cells themselves on the plate. Now, some bacteria, not all species of bacteria, some bacteria species have the ability to break down red blood cells, and Streptococcus species are really good at this. Inside of every single one of your red blood cells is hemoglobin. And inside of that hemoglobin molecule is iron. And these Streptococcus species need large amounts of iron inside of their for their metabolism to function properly. And that's why they have a hard time growing on nutrient agar. It doesn't have the iron in it for them. Well, anyway, what they're going to do is they have chemicals that they produce called hemolysins. Some of them are called streptokinases. It just depends on the book you find. Hemolysins, hemo meaning blood, lysins meaning split. So hemolysins that they will release outside of their cells. They're exotoxins. They will release an exoenzyme, not toxin, exoenzymes. They will release an exoenzyme outside of their cell, those hemolysins. And those hemolysins will come in contact with the red blood cells and do something to them. It will break them apart, poke holes in them, and things like that. And what you're looking at, or what you're looking for, are the different types of colors, different types of reactions that are produced by the hemolysins coming in contact with the red blood cells themselves. Okay, so the red blood cells come in contact with hemolysins, and hemolysis occurs. Blood lysis, splitting of the blood. Hemolysis will occur, and that hemolysis comes in 3 different forms, and that's the differential aspect of this blood auger. It's used most specifically for streps, and you diagnose the different species of streps based on how they hemolysize the red blood cells, how they can break apart the red blood cells. Now, the very first type of hemolysis is called a beta, and you can see this one here. This is a beta hemolysis, as well as this one. So beta hemolysis, these particular species of staph or strep or whatever it is, Streptococcus pyogenes is a very common one that does this. Streptococcus or Staphylococcus is hemolytic too. Staphylococcus aureus, you shouldn't be putting it on here anyway. But if you do on accident, this is what it will do. E. coli, a couple of other species, some species of E. coli are beta hemolytic. It just depends on the species. Beta hemolysis. So what's going to happen? Species that are beta hemolytic. These particular species of bacteria produce a very strong hemolytic enzyme. That very strong hemolytic enzyme is going to be released outside of their cells. It's an exoenzyme. That exoenzyme will come in contact with the red blood cells inside of the agar and break them apart, shred them to pieces, essentially just blow them up. And when they're blown up, the red color from the hemoglobin is going to be destroyed as well. The hemoglobin will be broken apart, and all of the iron will be stripped out of the hemoglobin molecules for use by the bacteria. So once the hemoglobin molecule is gone, that's what makes blood red, by the way. Once the hemoglobin molecule is gone, the red color of the blood cell is gone. And that's what you're looking at here. These particular bacteria, their hemolysins, have gone out into the agar, blown up all of the red blood cells that they've come in contact with, destroyed them entirely, removed all of the red color from this place in the agar. So like I mentioned earlier, this is nutrient agar plus blood. So if you have nutrient agar plus blood and you remove the blood part, you're left with a nutrient agar color, and that's what you're seeing here. So these guys have destroyed all of the red blood cells found in that agar. Very strong hemolysis. These guys will do the same thing inside of you. Streptococcus pyogenes causes strep throat. It shreds the cells at the back of your throat, and that's what causes all the pain. It's trying to get the iron from your red blood cells. So beta hemolysis. The second type of hemolysis is alpha hemolysis, or alpha hemolytic. So alpha hemolysis, these particular species have a weak hemolysin, a very weak form of hemolysin. What it does is it kind of just pokes holes in red blood cells. I mean, if you can kind of imagine stabbing a water balloon real quick and real fast, it's going to blow up, but that's what beta hemolysis does. Alpha hemolysis is more like taking a water balloon and poking it with a bunch of little tiny needles. The water just kind of slowly squirts out real slow, but the balloon itself doesn't pop. And that's what happens in alpha hemolysis. This particular species of bacteria, whatever it is, has a weak hemolytic enzyme. That enzyme comes in contact with the red blood cells, and it's just kind of going to poke holes in the cell membranes, kind of just weaken the cell membranes, kind of make the red blood cells kind of oozy a little bit. So all the good stuff inside of the cells kind of oozes out really slowly. It doesn't pop, which is why the color is still there. And if you've ever left a steak out on the counter too long, or you've seen meat that's been left out on the counter too long, it kind of turns brown. And this is kind of the exact same thing that's going on here. Hemoglobin, when it comes in contact with UV light, turns brown. It oxidizes, it rusts. And that's what happens here. These hemoglobin molecules have been popped from their red blood cells. They've been oozed out into the environment. The bacteria hasn't been able to eat them all quickly. They've come in contact with the UV light, and they turn this kind of a green, yucky, brownish color. And that's alpha hemolysis. You can't see through this plate. This beta hemolysis, you can see completely through if you were to hold it up to the light. This one you can't. It's just kind of a funky, greenish, yucky grossness. And this is the same thing that happens to meat on your counter. You leave it out too long, and it gets that kind of brownish yuck to it, just because it comes in contact with UV light and oxygen. Now, the last type of hemolysis is gamma hemolysis. And gamma hemolysis is a lack of hemolysis. These guys lack hemolytic enzymes altogether. They're going to be eating the proteins that are found in this agar, so they lack that hemolytic enzyme to break down red blood cells. Like I mentioned, this is nutrient agar, so they're going to eat the proteins that are found inside of the agar. They can't break down red blood cells, even if they wanted to, they just can't. So they're not going to have any change whatsoever. You're just going to have bacteria growing on a nutrient agar plate that just so happens to be red. They can't get rid of the red blood cells even if they wanted to. So this is gamma hemolysis. Beta hemolysis, very strong form of hemolysis. You can very clearly see the bacteria have removed all the red blood cells in that area that surrounds them. Alpha hemolysis, sort of a weak hemolytic enzyme, pops the red blood cells, they oxidize as they come in contact with UV light. Gamma hemolysis, a complete a complete lack of hemolysis. These guys just grow on a nutrient agar plate that just so happens to be red. Non-selective media, but it is differential based on that concept of red blood cells. Okay, so this is also a gram-positive agar. It's used mostly for gram-positives and specifically for streps. But it is something that most hospitals will just start bacteria on just because it's so easy to grow things on. Because it is for fastidious organisms. So if you have a strep, it's just easy to throw them on here and start going. Or something that you might think is fastidious, it's just easy to throw it on a blood agar plate and kind of skip the extra growing if it doesn't grow on a nutrient agar plate kind of thing.